This is something that quite a number of us will sort of bump into at some points of our career. And Fabrice Cordelière works at is a research engineer at the University of Bordeaux, and he works at the Bordeaux Imaging Center in France. And he's going to talk to us about colocalization now. Thank you, Fabrice.
Okay, thank you for having me. So the thing I did is really a digest of colocalization, focusing only on some of the quantifier and indicators that are the most commonly used, and putting aside everything about preparation of the colocalization analysis, but just giving you some warnings. Let me switch my presentation and then share it if I can manage. Okay, let's have a look at this. Let's share that. And now I will try
We
to
can see
go.
your screen.
Okay, can you see it in full? Is it okay on your side? Everything is visible?
Good.
Yes.
Perfect.
Good. So let's have a look into this black box that we called colocalization. And why? Why should we quantify colocalization? Because our eyes are sufficient. Look at this nice picture extracted from the ImageJ website. You see yellowish patterns. And of course, wherever you've got yellow, you've got colocalization. Do you all agree on that? Well, no, you don't agree because you know that there is a trick, and here it is. If you look closer, you will see that this is just the way you perceive the image that makes it look like yellowish. But if you zoom in, you will see that the two signals, the red and the green, are well separated. So this is why we can't trust our eyes. So we need to find ways, to find methods in order to analyze and quantify colocalization. But before we jump into quantification or characterization, we must make sure that we are all talking about the same thing. And actually, working on the facility helped me quite a lot trying to find definitions for what people think colocalization is. And in fact, there is not a single meaning for all users. Colocalization might be a lot of things. It could be, as depicted here, co-expression. You have a referential. This referential is, for instance, your cell, and you want to know if the two proteins are expressed within the same cell. But look at this example. Some of my users will talk about colocalization in this situation, even though you've got one protein in the nucleus and the other one, which is in the cytoplasm. You don't have any matching between the two proteins. You just have two actors which are in the same place, and you must define this place. This is why it's really important when starting to work on colocalization, first to define what you're talking about, and then to define what is your reference, what is the referential. At some point, this reference might be the resolution. For instance, here you've got co-occurrence, meaning that the two proteins are at the same place. But what do you mean by same place? Because of course, if you are taking the crappiest objective, everything will be colocalized. If you go to methodologies such as super resolution and so on, you might not be able to colocalize the proteins of interest the same way you did with a low resolution approach. So this is why saying that I've got co-occurrence, the two proteins on the same structures, this is something that is true, but only for a certain resolution. And actually, when you're working on colocalization, the conclusion might not be that the two proteins are at the same location, But the conclusion might be, knowing the current resolution, I'm not able to separate the detection of the two proteins. If you keep that in mind, you will see it will be a total game changer. So co-expression, co-occurrence, you might also have correlation. In this kind of situation, you're expecting some link between the intensities from both channels, from the concentration of your two proteins. You're expecting some relationship between the two, but you might have proteins of interest on the same spots with no link between the intensities. So this is why you've got to distinguish between co-occurrence and correlation. Finally, sometimes colocalization is co-distribution, meaning that you may not have all the proteins of interest from the two sets precisely at the same spot or linked in terms of intensity, in terms of concentration. But one pattern, for instance, of the green protein, will influence the localization of the red protein without having a perfect match in terms of intensity or location. In this kind of situation, you will more likely call it co-distribution, not co-occurrence, not correlation, co-distribution. So you see colocalization, one word, and at least four meanings. So this is why when starting a colocalization study, you must have in mind what you're expecting, at which resolution or at which level you're working. Then let's talk about the workflows. When you want to start looking for colocalization, characterizing colocalization, you must go through different steps that are depicted here. You must make sure that you have checked the data integrity. Sometimes you may have to do a bit of pre-processing. Then you've got a crucial step, which is choosing a reporter or metric. Then it's not even finished because once you have numbers, you must compare them in order to draw conclusion, to interpret the kind of output of the metric that you have. And finally, you will have to repeat this to assemble this into a workflow to apply it to a large data set. Let's quickly have a look at checking data integrity. So this is where I summarized everything on the slide and not going much into details because according to what Sylvie said, you have had or will have some lectures about that. Of course, you must make sure that what you're detecting is actually a pure signal, that you're detecting in the red channel only the red signal, in the green channel only the green signal. So you must be careful about bleed-through and also about cross excitation that might occur depending on the system you're working on. So really make sure that you've got pure signal in each of the channel. Of course, and especially if you're talking about co-occurrence or correlation, you must make sure that when you're expecting the two proteins to be at the same location, they are indeed at the same location. And you've got plenty of parameters that can affect what is called co-registration, such as some aberration in your microscope, some misplacement of the filters in the microscope. And if this is reproducible, maybe you will be able to correct for it, and we will see it later in the pre-processing. But you must make sure that you've got co-registration or that you have a way to go back to co-registration. And of course, resolution. Resolution is knowing your resolution is knowing your limit. So you may draw a conclusion out of your colocalization tests, and this conclusion will be, I cannot separate the two signals, the channel A and B, but knowing the current resolution. Maybe with a more advanced microscope, with a higher resolution, you might be able to see that your two signals are not colocalized. Without good images, there is no point going any further. And this is quite important. In image processing, we are not magicians. We are just dealing with the kind of data we are collecting. So if we don't get a good data set, well, there might be some little correction that we might do, but we can't create signal where it's not. We can't correct everything. So it's better to go back to the bench, prepare new samples if you've got problems with the dyes and so on, or talk to facility people or check the microscope on the acquisition part. Well, if generally speaking, when on the facility I don't get the quality check down, if the images are not good, well, as Mamaru would say, I'm asking my users to sashay away, and I will check with them and make sure that they've got pretty good data before resuming the analysis. About the pre-processing, there are many things that we can do, but once more, it's better to have good images. For instance, some corrections about bleed-through and crosstalk. There are methods that exist in order to reattribute to the proper channel the signal that is bleeding through onto the other channel. Chromatic shift and the co-registration might be corrected, but once more, once you've got pixels, you can shift one image relative to the other, but by blocks of single pixels. So it's always better to go back to the microscope where you've got a higher flexibility. In terms of background and noise, it's better to have good images with a nice signal to noise ratio rather than using filtering that might impair the resolution, or denoising. And denoising, especially, don't use it as a black box. Just try to think about what is going on and how it could affect your analysis. In terms of resolution, deconvolution is a good way to go, where you, knowing how the microscope will impair your image, you may try to reverse the process. But once more, deconvolution should not be used as a black box. And talking about black boxes, machine learning, this is quite exciting nowadays. But once more, make some tests before using anything to try to restore the data or whatever. Finally, a crucial point in pre-processing is being able to differentiate the pixels belonging to the background from pixels belonging to the object, so that you group the pixels belonging to the object into objects that then later on you will be able to analyze. And well, I guess that most of you already know about simple thresholding, but is it a good way to go, knowing that later on we will work on intensities? Well, it depends. And so thresholding might be an easy way to go, but not the only way to actually do the segmentation in order to isolate, delineate the objects, and so on. Now, let's step up to choosing a reporter or a metric. Is it okay for everyone right now?
Yep.
Okay. So let's go to choosing a reporter or a metric. Well, you've got many authors that have been working on that kind of subject, trying to have a look at what was going on in the field and separating all the reporter and metrics into subgroups. So we did it with my former colleague, Suzanne Bolt, and we decided to split the field of reporter and metrics into two big groups, the indicators and the quantifiers. I must say that some other authors have used different strategies, have had other point of views, but I will present what we've been working on. So indicators is simply a value that is computed and that has two extreme values, a minimum and a maximum value. But well, this is not a, this is a reporter of colocalization. This is not something that will give you the amount of colocalization that you find within your sample. Therefore, if this is an indicator relative to a range, you can draw pure conclusions from the extreme values. It's easy. If I've got the maximum value, well, I've got the maximum of colocalization. If I've got the minimum value, there is less chance to have colocalization, or maybe I've got exclusion and so on. But what about the values in between? Well, taken alone, this value relative to a range, unless you've got extreme values, it will be hard to interpret, and you will need to have at least a control or a reference, something that you can compare your value to. On the other side, you've got quantifiers. Quantifier is a reporter for a physical overlap. It's a reporter for the amount of signal that is engaged into the process of colocalization. So it can totally stand by itself. You've got a value that generally will range from 0 to 100%, giving you the amount of colocalization. So of course, if you've got a single experimental condition, you can interpret it. If you've got multiple, you can compare them and so on. But you don't have this uncertainty for mid-range values that you may have with the indicators. But we will see at the end of the talk that there are tricks that can be used in order to generate, even though you've got only a single experimental condition, to generate a reference value. So about the indicators, the most commonly used one is the Pearson correlation coefficient. Basically, what you will do is take all the intensities that you've got on both channels. You will take the pair of intensities, and the pair of intensities for a single pixel located in the same place, you will take the two intensities and put them on a plot using intensity of channel 1 as X and channel 2 as Y. You will end up with this kind of graph, which is called a cytofluorogram. And surprisingly on this one, you see a perfect line. This is due to the fact that I've been using twice the same image. So I end up with this line in the middle of the graph. You see that depending on the situation and especially the amount of noise, the dots, the cloud of dots will spread, allowing you to see a bit better the red line here, which is the line that is the more likeliest to represent the linear relationship that you expect between the two signals. Now, from that, you can compute this parameter, the correlation coefficient, that tells you how good the line here describe this population of values. So of course, if you take twice the same image, the line is just perfect. You've got the perfect relationship and you end up with a correlation coefficient of 1. if you've got a spread, and the more spread you have, the less the value of the correlation coefficient will progressively drop. So, but you see that there is, even though the coefficient is dropping, there is still a structure within this cloud. And you may think that, well, it's a line is good enough to describe this cloud. So you have correlation, but you see the value is quite low in between no correlation that you have when you have a coefficient of 0 and pure colocalization when you have a value of 1. So this is why I always said, well, taken alone, and especially only computing the value, it's hard to interpret the correlation coefficient. And moreover, the thing that nobody says, nobody's checking, is the correlation coefficient supposes that the distribution of intensities for channel 1 and channel 2 follow a Gaussian distribution, which is rarely the case in microscopy where you've got Poisson noise. What if instead of having this perfectly linear with a certain spread distribution between the intensities of channel 1 and channel 2, what if I get this kind of distribution, nonlinear? Well, there is a trick where instead of taking the individual values, you will take the rank of the value. This one is the first value. So I transform its intensity into rank 1. This one might be the 10th value that I've got on the range. So I place it here on the 10th tick within the axis. And this way I can linearize the signal. I've linearized the signal and I can use now the Pearson coefficient, sorry. And you see that it makes a huge difference. So once more, when using this kind of coefficient, you must make sure that the relationship that you get is actually linear and that you can use directly the Pearson coefficient. If not, well, there are some tricks, but once more, it's one trick that is suitable in this kind of situation, but not in all situations. Well, now we've seen an indicator. Let's have a look at the quantifiers. Quantifiers, I only need a single experimental condition to draw conclusion, and it directly gives me some hints about the amount of signal or the amount of overlap, physical overlap that I've got between the two signals. One of the major actors in the field is the Mendel's coefficient. Well, the Mendel's coefficients, because you've got two of them. In its original definition, the Mendel's coefficient is quantifying the amount of colocalization, the amount of signal that is involved in the process of colocalization. What you would do is find the intersection between the two signals, extract the intensity that you've got here engaged in the process of colocalization, and divide it by the total amount of signal you've got for one channel. This will give you M1. And you would do exactly the same for the second channel, meaning the part of the green channel that is engaged in this process divided by the total amount of green signal. So here you've got a percentage of signal involved in the process. Later on, there was two additional Menders coefficient published that were called thresholded Menders coefficient or modified Menders coefficient, where instead of looking at the intensity that is involved in the process of colocalization, you look at the area of signal that is involved in the process of colocalization. This way, you are independent of the intensities, whereas here you rely on the intensity. So here you are not expecting any correlation, just a match, a coincidence between the location where you find both signals. Talking about quantifiers, you've got more advanced methods that will rely on first identifying the pixels belonging to the object and grouping them into objects, and then making some measurements on the objects. Measurements that will lead to, for instance, for the red object that I've got here, defining where its center is. And its center might be geometrical, or it might be also based on intensity. Then you would apply a criterion saying, I consider that two objects are colocalizing if the center of one object is falling onto the other object. So in this specific situation, you don't have colocalization. In that one, you see that the two objects are overlapping, plus the center of the red object is falling onto the green object. So you would count this as one event of colocalization. The problem is that it might not always be a problem. If you've got a donut-shaped object, its center will be there, and you see that the center of the red object is falling onto the green object, although you don't have any overlap between the two signals. So you see that depending on how you define colocalization, you might, if you're not interested in this kind of event, you will have a false positive. But at the same time, if you want to know if your signal is on the outside of vesicles, you might combine several metrics. The one about the overlap, saying you don't have any overlap, but the center of one channel is falling onto the object from the other channel. So you see, it's important to have in mind how the metrics are working and adapt the picking of the metric depending on what you want to see, or even try to imagine new metrics, because this is a counter example for colocalization, but a really good example about co-distribution. So I told you this was a quantifier. How does it work? Well, simply by counting the number of events of colocalization. Maybe dividing the number of events of colocalization, dividing it by the number of objects you've got in channel A and in channel B. So you see plenty of possibilities. Another flavor of colocalization, you might this time try to summarize your objects by their centers. This is a shortcut that you may take, especially if you've got objects that are really small relative to resolution. It might be easier to work only on the center rather than trying to delineate really tiny objects that are hard to isolate. And then define a metric saying, okay, well, if the two centers are distant by a length that is below the optical resolution, well, then I will conclude that I'm not able to separate them. And as a biologist, I would say they are colocalized. But as an image analyst, I would say I'm not able to distinguish the two objects. Keep in mind this because it's super important. In biology, we go straight to a conclusion, but this is some kind of a shortcut. Now, comparing, interpreting the data. So you see, if you've got quantifiers, this is quite easy because you're reporting, the reporter has a direct meaning. But if you've got indicators and if you don't have anything to compare it to, well, it might be tricky. Therefore, you might choose several strategies if you've got a single experimental condition and nothing to compare it to. The first thing is to take the same content, same image, but twist it a bit. Here, I've got image A, image B, and the overlay between image A and B, and this Pearson correlation coefficient. Well, I will take exactly the same content, same image. A and D are the same. B and E, it's the same content, but not at the same location. I've been rotating the B image 90 degrees right. Yep. To get E, then you see the overlap. I don't have that many structures overlapping, and the correlation coefficient has dropped, meaning that yes, indeed, when I looked at the raw data, there was a correlation, a tiny correlation, but it was there and it was specific, not random. So this is a first strategy. Of course, if you've got quite a dense situation, even if you rotate, you will always have some overlap and a risk to have a correlation. So this kind of strategy is okay-ish in case you've got sparse objects. Another strategy is to translate one image relative to the other. This is exactly what you have here on the two images. Here you've got the displacement, and the graph is presenting as a function of the displacement, the correlation coefficient. This graph is called the cross correlation coefficient. You see, as we are coming closer to a zero displacement, the correlation coefficient is going up and it's going down on the side when I've got a huge displacement between my two images. So this means that indeed, I'm starting with this zero position where I've got the maximum of correlation, and going in both directions, I'm losing correlation, meaning that the correlation is specific. To the opposite, if I've got two signals that are excluded, one excluded from the other, the correlation is minimal when I've got no displacement, but as the mitochondria are entering into the nuclear domain, I have some correlation in both directions. So this is another way to say, okay, look, my coefficient might be low, but it is specific. Be careful about this kind of methodology. Of course, if you've got repetitive patterns, if you're working on muscle, for instance, well, you may have periodical bumps in the graph. So this is you just have to know it in order to be able to interpret the kind of graph you get. Finally, last strategy is randomization. Randomization is a bit like the first strategy, but a bit more complex. Instead of rotating, the thing you will do is cut out one of the two images into small pieces. You take all the small pieces, you put them into a bag, you shake the bag, and then extract the pieces of images, and you reglue them all together. And you end up with a different image, but with the same content. From there, you can compare the randomized image to the original image from the other channel and compute the correlation coefficient. You did, let's say, 5,000 times, and you might be able to plot the distribution of the Pearson coefficient that you get randomly. This is a distribution, and this is the correlation coefficient that I get on the two original unprocessed images. And you see that this value is far away from the distribution that describes random correlation, meaning that indeed it might be only 0.6, but I've got correlation here. A bit of warning about interpreting. Same value might reflect different experimental situations. You see roughly here, one third to one half of the green signal is involved in colocalization. Half of it is involved. Yes, but look, here I've got one volume. Here I've got two different objects. Here I've got a big object. Here I've got a small object. So putting some numbers on colocalization is good, but characterizing colocalization is good. But maybe here, to differentiate the different cases, I would also present a distribution of the size of the objects to say, I still have the same proportion of colocalization, but the most important parameter in this study is not colocalization. Well, it's a bit colocalization, but also the change in size and distribution of my signal. So always keep that in mind and always go back to the image and try to guess the kind of additional metrics that could help in interpreting and drawing conclusions. So when dealing with colocalization workflows and so on, always ask yourself, what type of colocalization method is most appropriate to my problematic? Are there any published methods that I can adapt or that I can directly use for my problematic? If not, well, don't be shy, be creative, build your own metric and characterize it, then use it and publish it. If something already published is good enough for you, fine. Just find the appropriate tool to apply this methodology. There are plenty of ImageJ plugins to address the colocalization measurement. So really feel free to use the one you're more comfortable with. Don't stick to the big standards and so on. Okay, so how long do I still have, Sylvie?
Oh, about 3 minutes.
About 3 minutes. And you've got the quiz as well, if I well remember.
Yeah, so that I think we will do in the next session, which is so we have some quizzes afterwards. We'll do that then.
Okay, so I will skip that one, which is a bit complex, and let's think about this kind of situation. You see, I've been doing some super resolution microscopy, something that I haven't been talking about previously. I've got detections, small crosses for the red channel, small crosses for the green channel. I want to characterize the colocalization between the two signals. What would you do? And this is, I won't be answering it. I will try. I just want you to try to think about the way you would do it. So if you've got some ideas, go ahead.
Guys, what would you do?
I'm just looking at the microphones. How would you handle this kind of problem? There are many ways.
Has anyone tried before colocalization?
So I see Christos.
I mean, briefly, shortly before we discuss, I think something similar that we can check the correlation coefficient and then try to do randomization and compare again just to see if what we get could be also random or if it's due to actual colocalization.
True. This is one way to go. But before that, look, I've got detection. So tiny little spots. Correlation is something that you would apply if you've got plain objects, and then you would try to correlate the signals from the two channels. So having tiny spots like that, it might be tricky to have correlation. So maybe we need a step before that.
Why would you say that it's trickier to have correlation with tiny spots?
Well, I place myself in a situation where you've got super resolution image. So basically, you've got localization. You,
Localization.
it's an image that has already been processed where you have detected all the PSFs. So of course, in this kind of situation, you won't have an overlap, a direct overlap between the objects. Therefore, if you want to use the regular methods, especially when working in super resolution microscopy, you must degrade somehow the image that you get.
Just have a look, Sadia. Did you have a question or a comment? Yes, please.
Yes, I had a question. There are a lot of websites that you can check subcellular, like co-localization of proteins. I was just wondering, like, how are they made? Based on what? The image analysis or RNA-seq data. So usually type in some, like, two proteins, and then they show the Pearson's coefficient. So I was just wondering.
Okay, so I don't know about all the websites and so on, but the easiest way to analyze colocalization for years and years have been Pearson and Menders. So basically, it's a coefficient that is easy to compute. So this is what is implemented behind. And unfortunately, sometimes this is not the best metric, and this might not be adapted to all problematics, especially if you're not expecting some relationship between the two channels, for instance. Is it answering your question, or am I off the?
I mean, it's answering and also not answering, because like this is image-based analysis, but then you also have I guess the data sets I saw were based on RNA-seq, so I would assume that they would be very different.
And unfortunately, but
yes, more objects, yeah.
I think it's different if we have small objects like RNA, like spatial transcriptomics, and if we have something that comes from SMLM, that is very different. So SMLM is this one one type of super resolution that Hans spoke about this morning, and there the image I think is very different. Very different. So it's a very special case that you're talking about now, where we have SMLM data, right?
Yes, and it's a special case, but just to illustrate that we've got some methodology in one field, and what it takes is just your imagination to transpose it into another field. So basically, if each single dot I convolve it, I transform it by the PSF just the way a microscope does, well, the image is not as nice as the previous one, but now I've got something on which I could apply the methodologies that I've been presenting earlier. I won't talk about tessellation, but working on distances is something that I've been presenting, and that could also work on this precise type of images. So, important message. When working on colocalization, have a look at the published methods, of course. But if you don't find an appropriate method, do not hesitate to be creative. Think about the uniqueness of your problematics. Keep your nerves, because sometimes colocalization might be tricky and so on, and take benefit of your talent and the imagination. So create your method, adapt it to your needs, and publish it. There is not big rules in colocalization, and except there are no big dogma in colocalization. Even though people are working with Pearson and Munders, these are only tools, and tools are made to evolve. They are made to ease the process, but if it's not easing your process, just go ahead and invent your own tool. Well, basically, this is a message that I want to
I think
share
it's
with you.
great. So what I find is interesting is that today our module is called Off the Beaten Track, and it's the second time today that you hear someone telling you, because Hans said the same thing. If this method doesn't work, just invent another one. So now I think you are considered super duper experts in microscopy. You can just go and do your own. No, but clearly this is what we do in science, right? We adapt things and we have to do that all the time. So that was very interesting. So thank you very much, Fabrice.
You're
and I would
welcome.
like to know, so please ask questions yes, sorry.
Just before the question, so it's been the first time that I give this lecture since I will try to phrase it well. So I would like to have a little thought for Susan Bolt, with whom I've been doing a lot of work about colocalization and who passed away 2 years ago. And this is the first time that I give this lecture since her passing. So little thought to Susan.
That is very kind. Is anyone, if you have any questions, or you can ask your questions now, and you're going to have to answer also something about colocalization and try to imagine how you're going to do it. Chen.
Yeah, thank you for your lecture. But I'm a bit lost because I know we will use correlation to find the relationship between two values. So how can you get the values? Because I think there are some methods that we like, for example, translate and randomize. We do some changes on the picture, but what did you measure when you do the changes, and how do you extract the values in correlation?
I'm sorry, I didn't quite get it. You want to know how we do for the cross correlation function?
Yes. So what are the two values you used in correlation?
You mean for the cross correlation, where do I start and when do I stop? Is that it? Or...
Yeah. For example, you do randomizer.
Okay. So you've got randomization on one side, and you've got the cross correlation on the other side. Cross correlation is this.
Yes.
I've got my two hands matching, and if I move one side or the other, my fingers are not matching. So the minimum and maximum displacement will depend on the size of my fingers.
Okay.
I will try to go until a point where I don't have correlation, back to the zero position where I've got the maximum correlation, and then on the other side to exit the finger. See what I mean? So this requires to have structures that have all the same size, so that you've got to know the size of one object, and this will define the amplitude of the displacement, the minimum displacement you should do in order to study the correlation.
Okay, I get it. Thank you.
And you were asking also about randomization.
Yeah, I think the principle is the same. Yeah, you just randomize them, and then you calculate the overlap.
But for randomization, you see that I've been taking big blocks.
Yes.
The blocks correspond to resolution in X and Y, simply because due to the point spread function, within a result, within an element of resolution, I've got a correlation, local correlation. So there is no need to randomize each individual pixel and place it on an image. What you need is to have blocks of image that you move around that correspond to the resolution.
But how can you define the length of the blocks?
The length of the block, well, you put some beads on your microscope that are below the optical resolution. You take an image. This image will give you a PSF, point spread function. So the size of a unitary object that is below the resolution, it will appear at the resolution. You define its size, and this is the size of the block that you will do during randomization.
You use the size of the point spread function as blocks. Yeah?
That's it.
Right. Yeah, that's the minimal that you can detect anyway. Right.
Thank you.
Welcome.
Good. Are there other questions for Fabrice?
I have a question. In which scenarios we would like to see colocalization? Just hypothetically.
In which
scenarios?
What kind of experimental situation
Yes.
am I confronted to for colocalization? Is that the kind of question? Okay.
Yeah.
Well, huge variety of situations. For instance, I've got researchers that want to know if when they express a gene, another one is expressed.
Okay.
In brain slices, for instance. So in this kind of situation, I will have a co-expression study where maybe the two proteins are not in the same compartment. One is in the cell, the other one is the nucleus. And I must adapt the strategy in order to count the number of cells where this phenomenon occurs. This could be if I'm expecting two proteins of interest to interact.
Okay.
If they are interacting, they might be in the same structures, or their location might be interdependent. And this is a kind of thing that I will use colocalization for.
For example, enzymes would be a good example. Any enzyme.
Yeah. The substrate and the enzyme.
Yes.
Could be a good example. However, if you're in the linearity of the association, you will be able roughly to use Pearson coefficient, for instance. But if you're saturating the active sites of your enzyme, well, you will reach a plateau. So in that case, you end up being in the situation that I presented, and you would have to use the Sperman coefficient.
Okay.
You see, once more, this is theoretical example, but this illustrates quite well two situations where the correlation might be used, but with some caution.
Also, it shows again the same thing that we have for microscopy, is that you need to have some sort of knowledge of your biology to be able to pick the correct technique, basically. So you need to implement the knowledge of the biology.
That's it.
Okay, thank you.
Welcome.
Are there any more questions? Well, if not, Fabrice, thank you very much.
Thanks for the invitation, and have a good time.
Okay, thank you. Bye-bye.
Thank
So
you.
now we're going to
Bye.
the other quiz. See you there.